So DNA replication, looking at our cell cycle, when we start, we're in G1 phase, we're growing, we're creating, duplicating organelles. We're starting this process of duplicating our cells to create two daughter cells. But after that G1 phase, we're entering S phase. And during S phase, the main thing we're doing is replicating DNA. We want the exact same copies of the same information, that same genetic code, so that our DNA is the same for our daughter cells. It would be an issue if we started dividing cells and they didn't have all the same stuff in them. We want all of that DNA to be the same. So today we're going to go over the process of that DNA replication. So thinking back to last week, just super quick, there are three pieces to a nucleotide. A nucleotide is a monomer of our polymer DNA, which is a nucleic acid. The three pieces, we have a nitrogenous base, that's our A, C, T's, and G's. Then those are bonded to our pentose sugar. In this case for DNA, that is deoxyribose. That is the name of our sugar. And that is covalent bonded to our phosphate group. The phosphate group and the sugars create our backbone. They're covalently bonded together because we need it strong and tight. Then those nitrogenous bases are kind of hanging off the end to sort of form those rungs of the ladder when they match up with another strand. So looking at our backbone, things are going to start to get a little bit more complicated because we're going to count carbons. We've done this before. It sounds a little familiar to when we're counting carbons in previous units. However, there's a very specific place we start so that we can figure out the orientation of our DNA strand. So DNA has a direction. There's what's called 3' and a 5' end. You can sort of think of it as like a beginning and an end, or a left and a right, or there's just an orientation. There's one side that's the 3' side, there's one side that's the 5' side. That little apostrophe after the number signifies prime, that word prime. So how do we know which end is which and why they're called this way? So looking at our diagram, it's a little bit small, but right here, we're going to start at our base. So our A, C, G, or T, our nitrogenous base here. That is connected to our sugar. In this case, our deoxyribose sugar. That is our first carbon. We're going to start our carbon counting there. So that's called 1' carbon. There's a little apostrophe, that means prime. We're going to start there. Then going clockwise, we're going to go to the second carbon, so 2' and 3'. In this example, the 3' carbon is on the bottom. That means the 3' end of the DNA is on the bottom. Continuing around our sugar here, we go to 4', and sticking up to the top is the 5' carbon, our last carbon here. Because the 5' is at the top, we're going to say the top of our DNA strand is the 5'. That's how we can figure out the orientation. 5' carbon at the top, so the 5' end of the DNA is at the top. The 3' end of the DNA is at the bottom because the 3' carbon is at the bottom. This can get confusing. As long as you start counting your carbons at where the base, the nitrogenous base, the A, C, G, or T, is connected to that sugar, count clockwise, then you can figure out the orientation of your DNA. In this case, this particular example, the 3' carbon is at the bottom, the 5' carbon is at the top, orienting our DNA with 5' at the top, 3' at the bottom. DNA, when we have our two strands connected by those bases, we have a structure called anti-parallel strands. Parallel because they're parallel, they're going the same direction. Anti or anti-parallel means opposite. So they are going in opposite directions. One strand has 5' at the top, the other has 3' at the top. One strand has 3' at the bottom, the other has 5' at the bottom. So they are going in opposite directions. They are parallel, but they're going opposite, meaning they're anti-parallel. How do we know which one is which? Well, we're going to count our carbons. So starting at our base here, we're going to start on the left strand here. We have an adenine as our base, so we're going to start there. It's connected right here, starting at 1', going clockwise to 3. 3 is on the bottom side here. We know that the 3' end of the DNA strand is going to be on the bottom. Continuing, 4, 5 is at the top. 5' end is going to be at the top. So our left side of our DNA strand is 5 to 3, top to bottom. Looking at our other half, starting at our thymine, because that pairs with our adenine, that will be our first carbon, our 1', going clockwise. 2, 3. 3 is at the top end. So we have 3' at the top. Continuing to count around 4, 5. 5' is at the bottom, meaning the 5' end of our DNA strand will be at the bottom. This strand from top to bottom goes 3' to 5'. They're going opposite directions because they're anti-parallel. The complementary strands run in the opposite directions. This is why counting our carbons is important. As long as we start at that base and count clockwise, we should be able to see which end is up, 5 or 3, and which end is down, 5 or 3. All right, let's look at some of the bonds that are happening. In our backbone, those phosphorus bases and our sugars, we have covalent bonds. Why do we want covalent bonds? They're strong, they're so strong. We want a strong backbone. What happens if we don't have a strong backbone? We're just gonna get all loosey-goosey and fall apart. We need a strong backbone. Our DNA needs a strong backbone to make sure things are strong and in order. Between our two halves, we have our bases, and our bases are ACs, Ts, and Gs. They are bonded together with hydrogen bonds. These are weak. Keep that in mind. As a review from last week, our purines are adenine and guanine, our pyrimidines are thymine and cytosine. They pair A to T, T to A, C to G, G to C. And in the AT pairing, there are 2 hydrogen bonds. And in the CG pairing, there are 3 hydrogen bonds. I'm going to glaze over this really quickly because it should be review from last week. Now we're going to get into how we copy our DNA. We're getting a little more into this new territory. So in our top right corner, we have one sort of look at it here. The blue or the darker color here is our original DNA strand. It's going to get split in half because we're able to break those hydrogen bonds very, very easily. So we're breaking it in half at those hydrogen bonds between our nitrogenous bases. Then we're going to pair our bases. So if A's always go with T's and T's always go with A's, we know what the other side needs. If C's always go with G's and G's always go with C's, we know which base we need to pair. And this way, we can duplicate and have 2 exact same strands of DNA. What does this mean? Let's break it down a little bit. At the bottom here, we have a set of progressive diagrams. So the very first one is our original strand. This is called a template strand. We are going to create our new ones off of this template. We're going to split it in half, break those hydrogen bonds, and start replicating it. We're going to add new nucleotides that match bases. So here we're adding on the light blue to the dark blue. So the dark blue is from the original template, and the light blue is the new stuff. Sorry, there is a, thank you for bringing that up, Zach. On the notes, there is a typo where it says, what does S pair with? That was a typo. I hit S instead of A, so it should just be A. So what does A pair to? It pairs to thymine. Thank you for bringing that up. I forgot to mention that. All right, and then, so looking at how we're pairing these things, A pairs to T, T pairs to A, C pairs to G, G pairs to C, we can create 2 exact copies. So if we look at the final pane here in our diagram across the bottom, our dark blue on the left side is the exact same as the light blue on the right side. And the dark blue on the right side is the exact same as the light blue on the left side. So we made 2 exact copies of DNA just by splitting it in half. This is called semi-conservative replication. Semi, because we're using half of the original strand for each new copy. And conservative, because we are conserving that code. We have the same original code and we have same original DNA, half of it, left over in our two new strands. So semi-conservative, semi because we have half, conservative because we still have half of the original, as well as conserving that code by pairing those bases together. All right, let's look at some DNA replication here. So in DNA replication, we have enzymes that run the whole show. Enzymes are workers in our body that without them, we can't get anything done. We wouldn't be alive. So we're going to focus on 4 main enzymes: helicase or helicase, depending on how you pronounce it, primase, DNA polymerase, and ligase. Notice that they all end in ase. If you see some sort of chemical that ends in ase, chances are it's an enzyme. And the cool thing about enzymes is the front part tells you what it does. 9 times out of 10, it'll tell you what it does. So you can figure out what that enzyme does and know it's an enzyme by that suffix ase. Really quick, looking at this diagram, notice that there are some of these bubbles. That is DNA separating. It doesn't all separate in one spot and then just go. It's much more efficient to open up spots and then connect them as they replicate. We'll get a little bit more detail into that. All right, so step 1. Before we can do anything, we got to break those hydrogen bonds. We got to separate those 2 halves. Helicase or helicase, its job is to do that, to unzip this DNA. It runs along the middle, breaks those hydrogen bonds, separates those 2 halves. Pretty simple. It's got an easy job there. Step 2. First part is primase. Here we go. It'll tell us what it does. Primase primes or gets things started. It's going to signal where to start this matching base pairing process. So it's going, hey, over here, we're going to start over here. I'm going to get a little bit going here, and then you can take over and continue to pair those bases. This green guy with the hard hat in our little Amoeba Sisters pictures here, I love them. They're very good. That is what's called DNA polymerase 3. They're the workers. They're the heavy lifters. They're going to do most of this pairing work. After the primer tells them, the little primase, hey, start over here, it's going to start matching and pairing those bases. Oh, cool. That's a T. I need an A. That's a G. I need a C, and so on. And it's going to start matching these bases to create a complementary strand. We're going to use that template, add bases to create a complementary strand so we have 2 exact copies of DNA. Now we're going to sort of simulate being a little polymerase. We're going to add new bases. So here we are, our DNA polymerase. And first up, going at the starting from the bottom here, we got our cytosine. What pairs with that? Guanine. All right, moving up, now I got adenine pairing with that thymine. And we keep going and we're going and we're going. All we're doing is pairing these bases, adding new nucleotides that match. Now, this is why it's important to know the direction or orientation of our DNA strand. DNA polymerase can only add new nucleotides on the 3' end of a DNA strand. So it has to work 5' to 3' direction. That means, looking here, we're adding nucleotides from the 5' to 3' direction. Notice the other side is upside down. It's anti-parallel. It's going the opposite direction. When we count these carbons, our nitrogenous base here, that's going to start as 1, 3' is on the bottom, so we can orient, 5' is on the top. That strand from top to bottom is going 5 to 3'. The other strand, when we count our carbons, 3 is on the top, 5 is on the bottom. It's going the opposite direction. It's anti-parallel. When we did our little animation here, I'm going to do it again. We can only add adding onto the 3' side. So we can only add going down in this example. Things aren't always going up and down. Sometimes they're going left and right. All right, I'm going to pause there. Questions? Repeats? Before we go into this last little bit. All right, so moving into the last little bit. So thinking about how DNA primase can only add on the 3' end of this strand. So going from 5' to 3' direction. That means we're going to have opposite strands. They're going in opposite directions. If we can only add going one direction, this might become a problem. Our helicase or helicase is splitting one way or the other, right or left. It's not going both directions simultaneously. This creates something called a lag. We're all familiar with lag. So we're going to have 2 different strands. So our top strand and bottom strand, right strand or left strand, whichever way it's oriented here. One will be what's called a leading strand. It's just going, it's chugging along, it's doing fine. And we'll also have something called a lagging strand, which needs to sort of catch up a little bit. So this is what it looks like. A leading strand, super easy. All it has to do is follow that helicase as it unwinds the DNA. It's going in the same direction as that helicase. It's 5 to 3' direction, same direction as the helicase as that DNA is unwinding. It's just a one-stop shop, just whoop all the way. Where we get some issues is in the other strand. Because our DNA polymerase can only add in one direction, 5' to 3', it needs to go the opposite way. But helicase is only unwinding in the other way. So we're going to get some fragments happening here. We're going to leapfrog. So as our DNA polymerase is filling in the gaps, it's going to reach that point where it started. So it's going to have to leapfrog back to where the helicase is unwinding, start again, go, oh, that's where I started the last time. Now I'm going to leapfrog again, go back to where helicase is unwinding and go, oh, I have to stop. And it's just going to go and leapfrog and leapfrog and leapfrog. And these little bits are called Okazaki fragments. These fragments were discovered by some guy named Okazaki, named it after himself, as are many things in science. So on this lagging strand where it's leapfrogging, it's creating these fragments. This is where I'm just going to leave it right here. This is where our last enzyme comes in. It's called ligase. Its only job is to glue those fragments together. We're going to have some little tiny gaps because we're starting and stopping and starting and stopping. And ligase just goes, hey, there's a gap. I'm going to whoop, glue that together. We're good to go. Oh, there's the next gap. Whoop, glue it. We're good to go. So ligase is just a glue. Here's a different representation. It's moving, which is always helpful. So on the top, we have our leading strand. It's just going in the same way that DNA is getting unzipped. One single continuous strand. On the bottom, we're seeing our lagging strand. It has to go in the opposite direction that the DNA is unwinding with helicase. You can see these fragments. The little green part at the beginning is our primase. The red part as it's filling in is our DNA polymerase. And as those fragments are getting glued together, that is our ligase. Here's a different look with our amoeba sisters. The helicase is unwinding our DNA. Our leading strand is on the top. It's just chugging along, going one continuous strand. On the bottom, in pink is our primase. In purple is our DNA ligase additions. Primase again tells our DNA polymerase where to start. And as it catches up, it has to leapfrog back to where the new stuff is as helicase is opening up this double helix for replication. These fragments, these Okazaki fragments, are glued together. It's not shown in this one, unfortunately, by ligase. All right, our last one here, we'll see our ligase in this one. The leading strand is not always on the top. It just seems to be the easiest representation out there. Sometimes the leading strand is on the bottom. But at the top, we have our one continuous strand with our leading strand. The bottom, we have our lagging strand in pieces or little Okazaki fragments, and our ligase is in there, gluing those fragments together. Comments, questions, concerns, clarifications? All right. Well, if you feel good, confident, have a wonderful day. I will see you tomorrow. If you want to ask questions, stick around. Come to office